Hey guys, Dr. B. This is just a exam review video to kind of help you to navigate stuff for Monday. Um, again, I'm not going to go over all these. There's plenty of coverage on this. I do have a short video on CPG islands that I have no idea why I didn't post, but I'm going to repost it again. So you want to check that little one out. That'll be an Edpuzzle one that I include. Very similar to what I covered in class last week on that. Epigenetic control and modifications. Realize that's kind of a little bit of a larger topic. It's obviously any of the non-genetic aspects of transcriptional regulation and how genes are transcribed and translated into proteins and things that would control that outside of the genetics. And I did give you a case, example of the Siamese cat's coat color. So obviously that's important. Going down the line, you definitely want to be aware of the different RNAs. Here, obviously talking about non-coding RNAs and what they do. RNAi, which is obviously RNA interference, hot air. All the RNA types, you want to be more of like a definitional sense. Like, do you know what they are and kind of what they might do functionally? So don't get crazy with them, but just be able to recognize one over the other. I'm going to skip the part on CRISPR for a second. So obviously with viral replication, this is very similar to what you may have had in 250. We want to obviously understand the lytic and lysogenic cycles and the differences between them. How obviously the lytic is going to be destruction of the host, and the lysogenic obviously will be incorporation of the phage DNA into the DNA of the bacteria, called prophage. And then, of course, as the bacteria continues to replicate, so does the phage DNA till some point when the bacteria is stressed. And then, of course, the phage DNA will erupt from the DNA from the host and then proceed to the lytic cycle, very much like the video that I made. Lovely. Very specifics here on HIV. This is not a course about medical diagnostics, so we want to really just focus on exactly what I wrote. Action of replication and the drug, what it does, AZT. DNA mutations and their causes, we spend some time on that as well, so you definitely want to know what kind of mutations can happen under a certain scenario, like ultraviolet radiation or chemical or whatnot. Moving down the line, deamination and depurination, I obviously talked about that also in class a couple weeks ago. And all the repair systems, very important stuff. Obviously, these go right along the line of CRISPR, especially the way that we can incorporate an oligonucleotide donor and the way in which the cell can incorporate that, just to be one example. Ames test, literally just know what it does and what it's used for. IGF-2 and VIN-3, just know what they are. Now let me talk about the red things. So the red ones are basically going to be large. These are going to be open-ended questions. So you're going to want to know how CRISPR originated and was discovered, the structure of the system, whatever the Cas enzyme and the guide RNA, how they're organized, and really what they do. So you want to include in there obviously a little bit of PAM sequences as well, protospace or adjacent motif, but spend more time on the fundamentals. Convince me you understand what CRISPR is, how it works, and what it does. You're literally in the middle of this lab now. And I highly suggest you actually look at your lab directions, your lab instructions I gave you for the CRISPR lab we're doing. Those first 3 pages of background are literally probably 90% of what you need for that. Then the last one, you're given a DNA strand pre and post mutation, and you decipher what type of mutation you would have. And so I'm going to now move to actual questions from the exam and show you what some of them would look like. All right. Now, as a reminder, your test is a paper test, but I'm transferring it from electronic, where I have I'm putting together the final paper version. But yeah, this is a question you're absolutely going to see. Very clearly covered this in an Edpuzzle that I made. So one of the easier ones, and so I'm going to show you a couple, probably 3 or 4 questions of different levels. So this would definitely be an easiest one. This one's a little bit more difficult, But again, because I, in the exam study guide, told you I exactly want you to know what VIN3 does, this question should be a little bit easier. But it's definitely more difficult than the first one because it's an example. So you need to understand the obviously the function of a mutation of this particular gene, and obviously in the nicest way possible. No, I'm not going to give you the answers to these. I'm giving you the questions, so you guys can go ahead and look them up. This one is a question that's probably one of the most difficult multiple choice ones on the exam, which is why I wanted to give it to you. And obviously you want to pause the video when I'm done talking about it and read it. But again, understand it's just testing your conceptual understanding here. You know, if you have an in vitro translation assay, which is an assay that's done outside of a cell, it's probably done in a tube. And obviously translation is going to be RNA to protein. And some of the amino acids were radioactively labeled so you could find them when they were incorporated into whatever the protein is. From this data, what can you conclude based on the amount of radioactivity? And so it's a really good question. And so I want you to take a look at that and make sure you understand that. So now I'm going to move to some of the open-ended stuff. This will be one of the open-ended questions. You're going to have a codon chart, of course. And in BIO263, you did the fairly simpler task of just identifying, obviously, which amino acids would be used based on a RNA strand. But in this case, you're going to look at what kind of mutation this is. So what I've erased here is a DNA sequence, and then the second part where the mutation, there's going to be pretty much all the same. There might be a letter different or whatnot, and your job would to be identify what type of mutation it might be: nonsense, missense, frame shift, inversion, something like that. So that's kind of what would be part of what was on the exam review that was in red. So this is worth more points because there's a few of these. And since this is your first exam for some of you with me, and first one with this course, which is a little different exam style than last semester, this is part of the question on CRISPR. It's a part of it. So I'm sure any of you can figure out that part of the answer, or part of the question, is going to be you telling me what A, B, C, and D are. There's way more to it than just that. So understand, if you just go by the exam study guide, the questions that I just showed you on messenger RNA and all of the DNA mutations, as well as the CRISPR, is literally between 20 and 30% of the entire test is those two, because they're extremely important topics. So hopefully that helped. And if you guys have any questions, it is Friday. There's a chance I might be able to respond to them this weekend, but it is the weekend. So, you know, I wouldn't wait that long. I also have class Monday morning, so please don't email me Monday morning. I'm not going to get back to you. So if you do have any questions, I'd like to get them to me as early as possible. If you are emailing me on Sunday, there's no guarantee I'll get back. So the earlier that you get these questions to me, the better. And I will see you on Monday.